Steve, I'm now inviting Steve Albarran up to the stage from Confident Cannabis, all the way from Palo Alto. Thank you so much for being here.
I'm really excited to be here.
Click down to advance the slides, and then the top star is the laser pointer.
Because we're working on some really cool things, and I'm really excited to show you. So my name is Steve. I'm the CEO and co-founder of a company called Confident Cannabis, and we are a software company, and we provide software to testing labs and capture that data and extract some really cool insights from it. So the topic of today is, what does indica and sativa really mean? So we all talk about indica sativa. Oh, I like sativas during the day, I like indicas at night. But what does that really mean, right? So let's look at it in a couple different ways. So first, a little bit about us. So our mission, the reason we started this company, is to help bring transparency to legal cannabis. And by transparency, I mean businesses knowing who they're trading with, and businesses and consumers knowing what they're buying, knowing what's in the plant, in that specific product that they're consuming or buying for their dispensary. So again, we're software for testing labs. We're nationwide. We're currently working with labs in Oregon, Washington, and California. We're onboarding labs in Colorado and Nevada. And as states legalize, we can provide services to the labs there too. And we're a team of 4 Stanford grads who started this company. We have a couple other people we're bringing on board soon. So basically, our software lets people order lab tests, they get the reports, and then they get the insights out of it. But for today's conversation, I'm going to use a couple different data sets. One data set of the lab data that we have, plus Leafly. For those of you who don't know what Leafly is a website that lets people rate their consumer experience. So they consume cannabis, they get high, and then they write about it. They say what they felt. They rate it on a number of different dimensions of how they felt. So we're matching those two data sets, the chemical composition plus how you feel. And for this specific presentation, I'm using 701 samples that have terpenes. Terpenes are really, really important. And a lot of people believe in this concept of the entourage effect. I believe in that myself, where all the different ratios together matter. I think somebody here was asking a question about the minor cannabinoids and other compounds and how they specifically react in conjunction. That's really important. It's not just about THC. So I'm using a much smaller set of the total of 100,000 samples that we have in our system. And big disclaimer up front, this is preliminary and exploratory. This is not enough to write a white paper, but it's just kind of cool and I wanted to share it today. So the common misconception is that there's basically one-dimensional paradigm, right? So you've got indicas on one side, it's what makes you feel stoned, it's the body high or nighttime. And on the other side, you have sativas, which make you feel high, it's daytime, it's a head high. And then you have hybrids everywhere in between. But cannabis is much more complicated than that. It's a false dichotomy. There's so many different things that you can feel. I mean, where do you put dry mouth? Is that daytime or nighttime? Where do you put anxiety? Nighttime, daytime? Where do you put, euphoric, right? Indicas make you feel euphoric. I like euphoria, right? So anyway, talking about things in this simple way of sativa versus indica is just not really giving cannabis credit for its diversity. So let's really dig down deeper. So let's look at indica sativa, see what it really means. There's basically 4 ways to think about it, right? There's morphology, which is the size and the shape of the plant, so the physical appearance of the plant. That's how most species in the animal and plant world are determined. They look at the way it behaves and the way it reacts, and they come up with a species. Then you look at genetics, right? So you can look at the DNA of specific samples and trace the lineage back to parents, and then try to infer some sort of classifications based on that. And finally, you have then you have chemotype, which is what these labs test. This is the chemical composition of the plant. So it's the cannabinoids, it's the terpenes, and it's a number of other compounds that you could test for. And then finally, the consumer experience, right? Which is probably the most important. You smoke it, you get high, what do you feel? Let's write about it, let's measure that, and let's classify that. So I'm going to go through 4 of these different ways of thinking about indica and sativa, and we'll see where we come out. So first on morphology, right? Indicas are shorter, fluffier, and they have wider leaves. That's a very clear distinction. And sativas have thinner leaves, larger, and they're taller and thinner, right? That's generally true, and that's probably how these labels came about. People looked at the plants, they looked different, they said, hey, let's call this a different species or let's give it a different name. So that's pretty accurate. Now, let's look at genetics. So there's a paper that came out, a couple of researchers in Canada. They said there's a moderate correlation between the genetic structure of marijuana and their reported sativa and indica ancestry, a moderate correlation. But the genetic identity of marijuana strains cannot be reliably inferred by its strain name or by its indica sativa status. Marijuana strain names often do not reflect genetic identity. So they basically said through genetic research, some researchers in Canada, that it's very unreliable to prove indica or sativa based on their genetics. Besides, most everything's been hybridized anyway, unless you go up to the mountains of Afghanistan and find an indica, or go to the hills of Jamaica and find a sativa. It's going to be really hard to prove what you're looking at on the shelf. Everything's been hybridized over many years. But so you can see here there's a loose correlation, right? So you've got the red dots, which are sativas, and you've got the blue dots, which are the indicas. And there's kind of a red cluster over here, and there's kind of a blue cluster over here, but it's kind of jumbled. So it's a loose correlation. It's not great. And then over here you've got, this is a genetic sort of spectrum here, and this is percent sativa here. And you can kind of see how it goes up and to the right, so there's a loose correlation there. But it's definitely not, foolproof, right? But then, this is from the same report. I'm going to go over here real quick. The red is the genetic sativa, and the blue is the genetic indica. And then here, you can't see it, is where they labeled it. So the researchers took these samples and they said, okay, this is 70% indica or 50% indica or whatever. And they kind of listed it with all these different strains. Well, 75% of the time, they got it wrong. So if you look at down here, so this is all blue down here, most of these are hybrids. In fact, there are a couple of claimed 100% sativas, and they have 0% sativa genetics, right? So 75% of the time they got it wrong, which is really interesting. So these things are just often mislabeled. So the punchline is genetics don't reliably define indica and sativa. So what else does? Right, so now let's look at chemotype, right? The chemical composition of the plant. This is what these analytical labs are testing. And this is where we're really interested, because at the end of the day, you're smoking a cocktail of chemicals, and that's what makes you high, so that's what we care about, right? So this shouldn't be a surprise, 97% of strains on the market today in the United States are high THC and low CBD, CBN. Now, CBN is a byproduct of THC, so you might not want to compare that to, other cannabinoids. But as you can see, everything is over here. Everything is high THC and low CBD. And there's a few high CBD strains here, which probably shouldn't even be labeled indica sativa, right? They're kind of in their own category. And then, just comparing THC and CBN. CBN makes you feel sedated, and it's kind of nighttime. CB nighttime is kind of the way I think about it, CBN. So the hypothesis would be, oh, well, maybe indicas have a lot of CBN. But that's not true. There's zero correlation, right? And besides, these numbers are tiny right here, right? So even if there was a correlation, it's negligible. And to Roger's earlier point, the ability to measure precisely these small quantities is just very difficult. So let's just say there's no correlation to THC and CBD to define indica sativa. But maybe potency. So maybe, let's say, THC is much higher for sativas than it is for indicas. That's a hypothesis. Well, let's look at that. Well, actually, no. The green is hybrid, the red is sativa, and the purple is indica, and they're all roughly the same bell curve. If they were truly different on THC, you'd see a spike over here for purple, and you'd see a spike over here for red, but you don't see that. They're all kind of mixed together. So again, potency doesn't define indica sativa. And let's try CBD, the other major cannabinoid. Again, same deal. There's a big spike right here at around 0.4, 0.5% by volume, by mass, sorry. So again, those don't define. Primary cannabinoids do not reliably define indica and sativa. So this is a quote that I found in High Times, which I really like. It's by Martin Lee. He said, so if THC levels are generally high across the board, and the other cannabinoids are present only at trace levels, what makes one strain different from another? And why does each marijuana strain impart a distinct psychoactive effect? There must be something else. All right? So let's look at that something else. Let's look at terpenes. So terpenes are really important. That's why I wanted to include them in this analysis. But by no means do I have an exhaustive list. There's, hundreds of terpenes that you could probably test for. Here I only have about 12. But I just summed up the terpenes, so total terpenes, really rough math. And I did the same analysis. I said, okay, well, maybe indicas have higher terpenes than sativas. But no, they all roughly have the same top of the bell curve in the middle. Now, there is one interesting thing here, is that there's no purple to the right. So just in this particular data set, which is not perfect, there are no indicas with terpenes above a certain point. I wouldn't necessarily draw any inferences from that. And then over here, I looked at what are the top terpenes. So I took all these samples. I said, what are the top 5 terpenes for indicas, sativas, and hybrids, right? And there's 6 here because I took the top 5 for all 3 categories, so I needed 6. The top is myrcene. We have beta-caryophyllene, limonene, ocimene, terpinolene, and A-bisabolol, okay? All of them are roughly the same. This is not statistically significant as a difference, right? These are basically the same values, except for terpinolene. Now, this is kind of interesting, right? This is the first place up to now that we've seen a significant difference between sativa and indica. Now, sativas have high terpinolene and indicas have low terpinolene. And coincidentally, terpinolene smells like pine. And in aromatherapy, pine is used to give you energy and wake you up. So that's probably the first place where there shows a difference. Sativas wake you up, right? But I don't think that, I think there's more to it than that. I don't think that this whole classification system, sativa equals high terpinolene, right? There's got to be more to it than that. But that's just really interesting. Let's put that in our back pocket. And then the other big takeaway is that there's a high degree of variance within each strain. So I'm using aggregation here. I'm taking hundreds of samples for indicas and hundreds of samples for indicas and just averaging them together, right? Now I'm going to do the opposite approach. I'm going to take one strain, one really common strain, Bubba Kush indica, and I took 10 samples out of 17 that I had in my database. And I measured the 3 top terpenes, beta-caryophyllene, limonene, and myrcene. If the strains were consistent, then you would see nice little plateaus here. Every strain would have roughly the same level of terpenes. You don't see that. There's huge variability. This sample has a lot of myrcene, and this sample has no myrcene, right? And they're all called Bubba Kush, right? Myrcene is pretty pungent. You can smell it when you pick it up. So there's high degree of variability within a strain. I did the same thing for a hybrid, the most popular hybrid, Girl Scout cookies. I did the same thing for the most popular sativa, Sour Diesel. You don't see any plateaus here. There's high degree of variability in each of these. So, I'm a millennial from Silicon Valley, so of course I have to use a texting motif in my presentation. So here I'm talking to Steve Stoner, right? He says, but wait, so if the cannabinoids and terpenes don't correlate with indica sativa, what defines an indica and sativa? The scientist replies, well, it's the shape of the plants, and sometimes they're genetics. But that's not getting you high. The chemicals are. But some strains actually make me feel up and others down. So I know there's a difference. Yeah, but it's not the indica and sativa designations that make you feel different. It's the chemotype of that specific batch. Sure, but I'm not the only one that believes in this. All my friends agree that indicas are for nighttime and sativas are for daytime. All right, let's take a look at what thousands of people say at Leafly. So again, Leafly is a review site where thousands of people review strains. I took the top 10 strains on Leafly. There's 2 indicas, Granddaddy Purple, and I don't know if you can see these numbers, but there's 1,000 for that, 1,000 for Bubba Kush, 4,000 for Blue Dream. So there's lots of reviews here. Blue Dream, Girl Scout, OG Kush, White Widow, Trainwreck, Sour Diesel for the sativas, Green Crack, and Jack Herrera. Okay? And then Leafly has a list of feelings that they can report. So when somebody writes a review on Leafly, they say they felt that or not. Okay? So coincidentally, every one of these top 10 strains makes people feel euphoric and happy. Of course. Marijuana is great. So that's common, right? That's pretty interesting. Everybody chose euphoric and happy for pretty much every kind of top 10 strain. But now let's look at the differences. So the indicas, as we would expect, show relaxed and sleepy, relaxed and sleepy. The sativas show energetic and uplifted, energetic and uplifted. And the hybrids show a nice mix in between. So far, the world makes sense, right? Okay, now, there's a caveat here. There's a little kind of nuance that people know the strain name when they're buying it, so they're expecting something, right? So green crack, I've never smoked crack, but I know it gives you energy. And so if I were to smoke green crack, I would expect to be energetic. So there's that bias there too. But let's put that aside for now. So far things make sense, right? Now, so Steve Stoner is really happy because he saw that page. He's like, aha, I told you. There was a correlation between how people feel and the strain names and indica sativa labels. Wait a second. Before you jump to conclusions, let's take a closer look at the data. So let's take those sort of hallmark feelings that we just talked about, and let's look at how many people said that in the reviews. So for Granddaddy Purple, only 44% of people said they felt relaxed. Only 42% said they felt sleepy. For Bubba Kush, only 42% they said sleepy, and 42% said relaxed. If you look at all of these numbers, they're all less than 50%. That means more than half of all the thousands of reviewers that smoked these strains and were given the option to select how they felt did not select that feeling. That means that if you're a bud tender and you get a patient coming in, says, hey, I want to smoke something at night to help me go to sleep, and the bud tender remembers this and says, oh, Bubba Kush, sleepy, try Bubba Kush. He's going to be wrong 58% of the time, right? Because these strains are highly variable. There's lots of difference batch to batch. You have to know that specific batch. Now, it's not to say this is all black magic and you can't measure this stuff and that, let's just throw up our hands. No, you can't draw these broad-based conclusions. Part of the reason is that the strain names are mislabeled. These plants are a function of their biology and their environment, so different growers have different processes, which creates a different chemical expression of the plant when it comes to harvest. Everybody's biology is different, right? You and I can smoke the same thing at the same time and feel differently, right? So we can't say, smoke this, feel that. It doesn't work. All right, so wow, Steve Stoner's convinced now. Wow, people really don't agree at all, do they? So if people are feeling differently, what determines how they feel? Again, it's the exact chemotype of that batch, as tested by a reputable lab. Reputable lab is important. All right, but how the heck am I supposed to know how much pinene I need in my OG Kush to relax? I can't understand this, I'm not a scientist. Well, with enough data, we can simplify the chemotype universe for you, and that's what we're working on now. So this is why we started this company. So how do I choose what to buy? Well, this is really, really preliminary. Don't take a picture of this screen. I mean, you can, just don't say that this is fact. But we're doing statistical modeling with our data, okay? And we've got some really smart people. We've got a couple of PhDs from Stanford, some from Harvard, which is awesome that cannabis has gotten to this point. And we're doing awesome modeling on this. Some machine learning algorithms, some neural networks, some crazy stuff that I fully don't understand. And they're clustering these strains. Because like Roger said, the truth is in the mean. So if you have enough data, if you have a big enough sample size, even though there's lots of variability, they're probably going to get to a middle, which is close to being accurate, right? So we're starting to do that, and we found that we're starting to find 5 key clusters. So it's not indica sativa. There's 5 key clusters. And these are the primary terpenes for those clusters, right? So cluster number 1 is alpha-phenylidrine, 3-carotene, terpinolene, alpha-terpenine. I'm not going to read these, but we can just start to think about things in that way. As soon as we're feeling comfortable with this and we kind of vet it with the industry, we can start to change the way we think and talk about cannabis and make it much more user-friendly for patients and consumers. Again, this is very preliminary. One big problem with where we are today is that we have a relatively small sample size. Even though we have 700 samples, it's not enough. We need a lot more. And so that's why we're working with as many labs as we can to get the data and do this math. We have a lack of minor cannabinoids. So that's a really big problem. There are lots of cannabinoids that are important that we're not measuring. And so my data set right now that I used only has THC, CBD, and CBN, but there's lots of other ones that are important. Mislabeled strains. There's a couple companies like Phylos Biosciences here in Oregon, another one called Mary Jean where they're doing DNA mapping and they're certifying a strain, so this actually is Blue Dream. One big problem in California, where I'm from, where there's almost no regulation, is that a grower might be growing, I don't know, call it Alaskan Thunderfuck, right? I just like that name. You know, he goes to a dispensary and tries to sell it, and the dispensary owner says, no, you know what? I really want Girl Scout cookies. Everybody wants Girl Scout cookies. It's the hot thing right now. I need to sell that. Come back if you have some. Comes back a week later, sells his Alaskan Thunderfuck, calls it Girl Scout cookies. Hey, I got some, you know? And so that's a really big problem, is mislabeled strains. And people are self-selecting the strain name when they're testing, so the lab doesn't know the difference. So that's a really big problem. There's a little too much market gaming with the strain names, so we have to verify that with groups like Phylos and Mary Jean. And then finally, imprecise measurements. There's some really good labs, but then there's some labs who are not as good, and there's inherent variability, so we just have to count for that. But this is what we plan to bring to consumers. This is sort of our vision, and that we hope that you guys help us get there, which is once we say, okay, it's not indica sativa. There's a lot more to it, but we can simplify it. You can't just say, here's, you know, 1.2 milligrams of terpenes. This is cluster number 5. And I bought cluster number 5 a week ago, and I really liked it. And now I'm going to buy number 5 again. And I might like 5 for a different reason than you like 5. But now I know every time I go to a store, whether it's in Oregon or Washington or California or Massachusetts, and I buy 5 number red, or red number 5, I'm going to get what I like, which is really cool because now you're finally measuring the exact cannabinoid profile for that specific batch and the exact terpene profile for that specific batch. You don't worry about strain names. You don't worry about indica sativa. You don't worry about, you know, different growers with different processes. You know what you're getting. And so that's kind of what we hope to do, to help people make better purchasing decisions, and businesses too. So if you're running an extract company, you need to measure your you know, the quality of your raw ingredients. You need consistency. Same way, you know, Coca-Cola needs to know exact sweetness of the corn syrup that they're putting in their beverages. You know, a good extract manufacturer who's built a brand on consistency and reliability with their consumers has to know the raw ingredients that's coming into their factory. So they're going to rely on this too, we hope, we think, we expect. But we're not there yet. We're early. We just started this company in March. Thank you. We just started this company in March, and we're signing up labs. We've got about 13 so far. We're just, you know, rolling that out. We need a lot more data. We need a lot more help from people like you guys. But we have to start somewhere. So this is one thing that we're doing. So Roger mentioned the Cannabis Transparency Project. It's really, really cool. They're using Washington publicly available data. Because it's publicly available, we have that as well. So Washington are the red dots here. This is a screenshot of our platform that labs and people who use these labs get to see. So if you guys test with OG Analytical next week, you'll see this. And the blue dots are California. And so this is dynamic, so you can hover over these dots and you can see a constellation of THC on the y-axis and CBD on the x-axis. And for the very first time, you can see what the world looks like state by state. And again, California does not have publicly available information, but we have labs in California, and so we aggregate and anonymize it. Each dot is a strain with at least 15 samples. So it doesn't go on the page if you have just one sample, because it's not statistically valid. We just chose the number 15. So now you can compare how, you know, Blue Dream in Washington looks like Blue Dream in Oregon or Blue Dream in California. And over time, we're going to add a lot more analytics to this so that people can really see how they're doing. So now growers can see how they're doing harvest over harvest, and they can track it automatically and see it in really amazing, beautiful dashboards. And then you can also see how you're doing relative to your peers. So this is something really interesting that we found. Northern California consistently has higher THC levels than Southern California. Oregon probably is a lot closer to Northern California. I think it's because of the sunshine. Sunshine's pretty good. Sunshine is good. But Southern California is catching up, right? So this is information that doesn't exist anywhere else and nobody knows, but we're able to show. And this is just available to people who use our platform, and we'd love to sign up as many people as we can. So again, my name is Steve. We're Confident Cannabis. We're a data company, and we're helping to bring transparency to legal cannabis. Any questions?
So everybody gets excited about the high THC, but as was brought up with another presenter, and you just mentioned it, with the entourage, it's such a complex plant with all these different compounds that have to work synergistically. Like Jeremy, who gave a presentation before, said, they're getting it to where there's such high THC that you may as well smoke Mirasol. And then I guess apparently with the high THC, it's creating more anxiety, more aggressiveness, and the CBDs then plummet, right, as far as the ratio goes? And anyway.
Yeah, you're absolutely right. So one of the really cool things, so I believe everything you said, and I think a lot of people who are really knowledgeable in the industry believe exactly what you said. A lot of sort of newcomers to the category or people who haven't studied the science as much, they just say, wow, high THC, I'm going to buy that one. But one of the cool things we're seeing is that there's this huge demand for higher CBD levels. So there was kind of this pendulum swing. So, I mean, let's face it, cannabis came out of the illicit market, and it was sold in conjunction with other illicit drugs. And so it was about, your bang for your buck. And so people want to get high, people want to get fucked up. And so people were intentionally breeding for high THC. But now it's going the other way. People are starting to become more connoisseurs. They're starting to, appreciate the nuances of it. And there's been a lot of research that has come out recently saying that, the CBD levels actually help regulate a lot of these negative effects like anxiety and aggression, like you mentioned, in the high THC. That's the reason why Marinol hasn't been very successful, because it's 100% THC. You need the other cannabinoids to balance things out. So we are seeing a huge demand for higher CBD. Dispensaries can't keep enough of it, at least the ones that we're seeing. So that's good. That's a good thing. The other thing, to your point, is that it's the ratio that matters, not the absolute value. Right? The ratio is what matters of all these compounds to one another. So when I showed you the clustering, these are not absolute values. In other words, limonene is not in cluster 2. So if a strain has high limonene, it's not in cluster 2 because it has high limonene. It has to do with the ratio to the THC, to the other cannabinoids, to the fentanyl. It's a multidimensional ratio because it's about the cocktail. It's about the proportions of one to the other. And that's what matters. And so that's what creates the high. That's what creates the effects. That's how we're doing the clustering, not an absolute value.
Yeah, I haven't heard much said tonight about growing something indoors versus growing it outdoors. That's one question. And then another thing, I think we need to breed marijuana that allows people to drive without having the police stop them, because a lot of people are driving too slow, and I'm hearing that that the police are arresting more people now because of marijuana. And then the final thing is, I think we really need to get right to the point and figure out which strains make us most creative to solve the world's problems we have right now, and also to create the 90% THC that Rick Simpson says is curing cancer, that extract.
So the driving question. Driving too slow is better than driving too fast under alcohol, but it's still, you shouldn't drive under the influence. There are a couple of companies who are trying to create breathalyzers. There's a challenge because because it's fat soluble, it's in your body for a long time, even when you're not high. It becomes difficult to measure if you're actually high in that moment. But there's some people who are trying to solve that. I know a couple of companies who are doing that. The Sunshine Outdoor, I'm not a grower. I don't know anything about growing necessarily, but I do see the lab data. And outdoor strains typically have more favorable profiles. So whether it's high THC or more terpenes, they often don't look as nice because they're outdoor. So they often don't sell for as high a price. But this plant has evolved in the sunshine, so give it sunshine. And then around the creativity, again, I can't recommend creativity because everybody's different. And so I think people have to find a cluster that works for them. So if number 5 makes them creative, they can buy number 5 next time. But number 5 is not going to make everybody creative.
Cool, thank you. Steve, great presentation. With the different categories here, what would you foresee the challenges of potentially using these categories for a regulatory-based labeling structure as far as conveying that information in different, right now, disjointed regulatory environments, but potentially in the future? What significant challenges with these different classifications do you see? And would the ability to use these different groups for regulatory labeling, do you see the genetic identification as far as strain positive ID being kind of along the same path, or timeliness, one or the other kind of hitting market first necessarily?
Yeah, so the regulatory constraints I think are more around consumer protection. I think, I might be wrong, you guys are talking to regulators probably more than I am, but I don't think that regulators necessarily care as much about consumers getting the high that they expect, and they care more about consumer safety. So they're more interested in microbials, pesticides, absolute potency. So how high are you going to get? Not are you going to be creative or take a nap? So I don't see too much pushback on sort of the way that people talk about. I mean, regulators aren't even coming down on strain names like green cracker, marijuana, right? So I don't know that they'll worry too much about the classification, the way people think and talk about it. But the labeling is really important. So, for example, this is just a mock-up. I just made this for this presentation, and I'm using the nutrition facts layout because we're familiar with it. But different states have different minimum requirements, right? In California, every municipality has a different labeling requirement. So one of the cool things that we can do with our software is that growers can have their artwork uploaded to our software, and we get a little piece of real estate where we can drop the lab data for that specific batch, and they can hit print. And all they have to do is select what region they're selling this product in, and all of the compliance is taken care of. They don't have to worry about it. We figure out all the laws once instead of everybody having to figure it out every time. So we're going to definitely comply. It's going to vary, though. Any other questions?
I'm really curious with the terpene levels.
Let me give you the microphone here.
You know, we're looking at these different terpene levels, and like for instance, I buy limonene as a cleaner, 95% food grade. Are you seeing adulteration? I mean, people saying, well, I want my terpene levels up, so I'll just soak it in the limonene?
So the question was, are people artificially increasing the terpene levels? I don't know, because I only get the lab reports. So if the lab figures it out, then they probably will change the number they're reporting, or I don't know, maybe not. If it's actually limonene, then I don't know, Bethany, I mean, Roger, I don't know if you can tell if there's a difference in it, if it's synthetic or not.
If it's there, it's there.
Right. I mean, if it's the same compound, it's there. So I don't know. I haven't seen it because most people don't maximize terpenes right now. If people clean their houses with THC, then people would definitely be dunking their weed in THC. But people aren't optimizing for terpenes yet.
I think that's why they're leading in Europe, because they don't have to account for each plant. If they have a low THC strain, they can grow that freely, and they don't have to apply for a permit. And of course, their hemp research and cannabis research for THC low strains, or even THC free strains, if something even exists, is much more prolific in Europe than here. Because here, of course, the grower wants to focus on the THC strain he or she can make the most money with. So that's really a big difference. And what we see in Austria and Germany and Switzerland are these CBD strains and high terpene, high terpenoid CBD strains that they want to develop. And now we have GW Pharmaceutical has just a couple of clinical trials in. The first one passed the first phase for a CBD preparation. And so in terms of clinical trials and CBD research, the Europeans are much more advanced because they didn't have to care so much about the prohibition of hemp as such. And I just wanted to ask you about that. To what degree the regulation or to relax those regulations would benefit cannabis research for THC low hemp strains?
Yeah, so that's a great question. The world definitely needs more cannabis research. I mean, this industry is happening. That's why we're here. We need to know more about it. We're still before dawn. There's a lot more to know. Europe is very different than the U.S. The U.S. is driven by free enterprise, and so businesses are kind of self-regulating like we are today here, and the businesses are advising the governments on how to set rules, and everything is sort of independently run, private enterprise. Whereas in Europe, typically, the government's the ones that's actually in the business of operating these cannabis businesses. So, for instance, in Italy, the Italian military is growing cannabis, which is kind of cool, I think. And so it's just a very different world. So they can, because they're the government, they can do whatever they want in their own jurisdiction. There is a lot of great research that's coming out from Europe, and people are publishing it, and we're learning from it. And then there's a lot of great research coming out from the United States that's getting published. The federal government in the United States recently broke down a few barriers to the ability for labs to conduct research on cannabis, which is a really, really great thing. I think that happened a couple months ago. So the trend is definitely going. These analytical testing labs, companies like ours that are collecting the data, we're adding to that.
Just offhand, I don't think all the terpenes that are in your clusters are psychoactive. So are you actually measuring potency with some and taste with others, or are they all determined to be psychoactive components?
That's a really good question. I wish I would have clarified earlier. The question is, are terpenes psychoactive, and if so, why am I using them to cluster? Or if not? The answer is kind of. So research shows that consuming terpenes in sufficient quantities does change the nature of the experience. So there is some truth to aromatherapy. It's not all hocus pocus. And so when you consume these certain fragrances and aromas, there is a physiological response. It's not necessarily psychoactive. So CBD, for example, is not psychoactive. That doesn't mean you don't feel it, right? Advil is not psychoactive. But if you have a headache, it goes away. So you feel it, right? So the answer is kind of. No, terpenes don't make you high outright, but they do change the nature of the high. And I'm talking about terpenes because I have that data, but there's a lot of other cannabinoids that I don't have that are probably really important. So there's just a lot more to it.
I find it really, really fascinating that in Colorado, for instance, the Stanley brothers are working with a low THC, 0.3% hemp. And it's working incredibly well for children, especially with the CBDs, with seizures and such. And 70 years prohibition and hemp being classified a class 1 substance has been put on the back burner. And it's really interesting that medical and recreational high THC are taking the lead, where 13 licenses were given to grow hemp. But now there's this fear of pollination of low THC contaminating the high THC. And that's a real fundamental question of how we're going to coexist with that.
Yeah, so as I mentioned before, there's a lot of demand for high CBD strains. My personal favorite is Canatonic and Harlequin and AC/DC. Those are some of my favorite strains because often they have some THC, but they have CBD, and it's just really balanced. I'm not a fan of the heavy sativas. But anyway, this is basically what you're talking about right here. So these are the high CBD, low THC. So this is probably, Charlotte's Web, for example, like the guys that you're talking about in Colorado for kids with epilepsy and to treat seizures. because it's not psychoactive. There's no THC in it, but it has high CBD and it's very medicinal. And so there's a whole group of strains here. This is brand new territory. This hasn't come out of the black market really because it doesn't really get you high, right? But it's really, really important. And so the line between, you know, quote, marijuana with low THC and quote, hemp is blurred. Some people use the terms interchangeably. You could consider this hemp. Hemp has CBD in it. But because of the way it's produced, because, you know, people tend to call it marijuana, I haven't heard anybody worried about the CBDs pollinating the THCs because typically the THC strains are kept in an environment where it doesn't get pollinated by anybody or by any plant. In fact, you have to determine the gender of the plant when it's you know, young, so that you don't have no males to pollinate the batch. Otherwise, you don't flower and you ruin the batch. So I haven't heard the pollination problem, but yeah, this is really, really important. There's a lot of interest here, and there's a lot of growth here. If I were a grower, I would definitely be growing one of these. There's good money to be had there.
Actually, the high THC are grown mostly from clones, so there's not the fear of having the male. Some go hermaphroditic, they go back, it will go to seed. But the hemp is grown on a large scale agriculturally, and it's grown to its full potential of, it's grown closely 4 inches apart for various, many thousands of uses. So it's grown it for the oil in Canada just across the border. So it has to go to the seed stage, which is where the oils are concentrated for full proteins and multiple uses for the oils. So that's the fear, is that since it is grown on an agricultural scale outdoors, that the pollen is going to contaminate the female clone plants of the high THC. And so that they're not having that problem in Canada because they're not concentrating on medical and recreational THC plants. So that's something that has to be addressed in this country, how we're going to work with that.
Thanks, I didn't know that.
We're just going to do one more question for Steve. Here, Kathy.
Just yesterday in the Register Guard, there was a letter to the editor from a man who said that there's a war against marijuana, and he was quoting the fact that Peace Health Hospital here has given a letter to their doctors that they not only can no longer prescribe medical marijuana, but they have to eradicate their records. And I wondered if you're seeing this other places happening. It's interesting, this has gone on just 2 weeks before the October 1 adult use is being legal. But what we need to do is come up with a non-THC strain so people can actually have their PSDS and all these other things helped without worrying about the medical establishment being threatened, because there's a $100 billion a year cancer industry in this business. We got to get rid of this pink ribbon nonsense.
Yeah, that's absolutely right. So these are the non-THC strains, and like I said, that's where the growth is. That's where I would invest. All right, thank you so much.